Welcome back to the deep dive. Today we're plunging into something really quite surprising at the crossroads of aging, blood health, and, well, cancer therapy. We're looking at a specific age-related mutation in blood cells. Historically, this mutation, definitely bad news, linked to things like heart disease, blood cancer risk. But now, the story's flipped. We're asking, could this supposed bad gene actually be a key player in making modern cancer immunotherapy work better against solid tumors?
It's a fascinating twist, isn't it? For years, we saw this as a negative sign, a red flag. But the latest research, the sources we're diving into today, they show how this mutation can transform into a powerful biomarker, predicting, believe it or not, better responses to treatment. So our mission today is to unpack that, look at the human data first, and then really get into the weeds of the cellular mechanics behind it all.
Okay, let's start with the basics then. The first term we really need to get our heads around is clonal hematopoiesis, CH for short. I think when people hear blood mutation, they might jump straight to leukemia. But CH is different, right? And why is it so common, especially as we get older?
Right, good question. CH isn't cancer itself. It's more like a consequence of aging within your bone marrow stem cells. Think of your bone marrow constantly producing blood cells. Over time, the stem cells can pick up mutations, just random copying errors sometimes. If one of those mutated stem cells starts to, outcompete the others and its descendants, the mature blood cells make up a noticeable fraction of your blood, say 2% or more. That's what we call CH. And it's surprisingly common. We see it in over 10% of healthy older adults, and even more frequently, maybe over 20%, in patients who already have solid tumors, which is interesting in itself.
Okay, so it's a sign of aging in the blood system. And within CH, there are different possible mutations. But we're zeroing in on one specific gene today, TET2. What does the TET2 gene normally do before it gets mutated?
Yeah, TET2 is actually the second most common gene found mutated in CH. Its day job is crucial. It's essentially an epigenetic regulator. Think of epigenetics as like instructions written on top of your DNA telling genes when to switch on or off. TET2 helps write some of those instructions. Specifically, it's involved in a process called DNA demethylation, removing certain chemical tags. When TGO2 gets mutated, that whole editing process gets a bit, messed up, especially in the myeloid lineage, the cells that become macrophages and monocytes.
So the quality control for gene expression in certain blood cells breaks down. And like you said, historically, this TET2 mutation and CH in general spelled trouble. You mentioned links to blood cancers like a tenfold higher risk, plus heart disease, stroke.
Exactly. That's the backdrop that makes this new finding so striking. Finding TET2-mutant CH used to just mean higher risk, more inflammation, generally a worse outlook. But the pivot here is that the new data points to TET2-mutant CH as a predictor of improved survival, specifically, and this is key, when patients get immune checkpoint therapy, or ICT.
Right. That condition only with ICT is critical. So a risk factor flipped into an advantage, but only under specific treatment conditions. Let's get into that evidence. You mentioned massive patient groups. Tell us about the clinical studies.
Yeah, the clinical validation is really solid here. The researchers analyzed data from, huge numbers of patients. We're talking 35,971 people with non-small cell lung cancer, NSCLC, and another 25,064 with colorectal adenocarcinoma, CRC.
Yeah, okay. Those are definitely large numbers, high confidence data, like you said. So what was the bottom line for the patients in those groups who had this TET2 mutation in their blood?
The main finding was crystal clear. Patients who had TET2-mutant CH lived significantly longer. They had better overall survival. And this held true across different types of ICT drugs, the common ones like pembrolizumab, nivolumab, atezolizumab. And here's the clincher that really points toward the mechanism, this survival benefit. It only showed up in the patients who actually received immunotherapy.
Okay, that's the crucial control comparison, isn't it? Because if the mutation just made the tumors inherently less aggressive, you'd expect those patients to do better regardless of treatment. So, just to confirm, in patients who got other treatments, like standard chemo but not ICT, their TET2 status didn't matter for survival.
That's exactly right. In the non-ICT groups, having the TT2 mutation made absolutely no difference to how long they lived. It strongly suggests the effect is all about the interaction between the immune system carrying this mutation and the way the immunotherapy drugs work. The mutation isn't directly weakening the cancer. It seems to be changing how the patient's own immune cells, specifically those derived from the mutated clone, respond to the therapy and how they influence the tumor environment.
Got it. So the TET2 mutation is somehow prepping the battlefield, making the immune system more receptive or effective when the ICT arrives. We know what happens in people. Now, the really fascinating question, how? What's the biology? Let's dive into the mechanism.
Okay, so to figure out how a blood cell mutation could affect a solid tumor in the lung or the colon, researchers turned to mouse models. It's the standard way to dissect these complex interactions. They essentially created mice that mimic human CH. They transplanted bone marrow cells carrying a TET2 mutation using the mouse version of the gene into recipient mice, and then gave those mice common types of tumors, like pancreatic cancer or melanoma models.
Right, so they've recreated the human scenario in a controlled lab setting. Who are the key cellular players they observed in the tumor itself?
Well, it turns out the stars of this show are the macrophages. These are immune cells, part of that myeloid lineage I mentioned, derived from the mutated bone marrow stem cells. And these macrophages tend to heavily infiltrate many solid tumors. What the researchers saw was really striking, especially after treating the mice with ICT. There was a significant change in the identity and function of the macrophages that carried the TET2 mutation.
A change in identity. What did they start doing differently? You mentioned macrophages often infiltrate tumors, but don't they sometimes suppress the immune response?
That's often the case, yes. In many tumors, macrophages can differentiate into types that promote tissue repair or suppress T-cell activity, kind of like a wound-healing crew that inadvertently helps the tumor hide or grow. That's what they saw more of in the normal or wild-type mice. But in the mice with TET2-mutant CH, especially after ICT, there was a big increase in macrophages, expressing MHC class II molecules on their surface. So MHC2-positive macrophages.
Wait, MHC2, those are the molecules immune cells use to present antigens, right? To show pieces of the enemy, like tumor fragments, to T cells. So they're becoming better antigen-presenting cells, APCs.
Precisely. They're essentially shifting roles. The researchers used single-cell RNA sequencing, a powerful technique to see what genes are active in individual cells to confirm this reprogramming. They found about 10 different subtypes of macrophages in the tumors. In the normal mice after ICT, the macrophage population shifted towards those tissue remodeling types. But in the TET2 mutant setting, there was an increase in what they called IFNγ plus macrophages, meaning interferon-responsive macrophages. These mutant macrophages turned up genes needed for presenting antigens via MHCP, like H2-DMA and CETA, and also genes for cross-presentation via MHCI, like PSMB8 and PSMB10. So your analogy works well. Instead of being the suppressive repair crew, the TET2 mutation seems to push them towards being an immune alerting signal unit, really good at showing the T cells what to attack.
That's a fundamental shift. So the mutation isn't just present, it's actively changing the job description of these key immune cells within the tumor itself, making them more pro-inflammatory, more immunogenic, less suppressive.
Exactly. And that shift immediately pointed towards a really critical signaling molecule, interferon gamma, or IFN gamma. We already know that IFN gamma signaling is absolutely crucial for making ICT work effectively. It's one of the main signals that tells cells, including macrophages, to put up those MHC flags and get ready for an immune fight.
Okay, so the hypothesis then becomes, does the TET2 mutation somehow make these macrophages hyper-responsive to that IFN gamma signal, like turning up the volume on the alert system?
That's precisely what they tested next, using macrophages grown in the lab, bone marrow-derived macrophages, or BMDMs. They exposed both normal, wild-type, and TET2-mutant macrophages, the IFN gamma. The wild-type cells, again, tended to activate pathways related to things like collagen organization, that wound healing profile. But the TET2-mutant macrophages, they strongly activated pathways involved in positively regulating T-cell-mediated cytotoxicity, things like a type 2 interferon response itself, and antigen cross-presentation pathways. They were clearly geared up for a fight.
So they didn't just react differently, they reacted more strongly. Was the response amplified?
Yes, that seems to be the key mechanical insight. The TET2 mutation appeared to enhance both the magnitude and the duration of the response to IFN gamma, give both types of macrophages the same IFN gamma signal, and the mutant ones responded more robustly and for longer. It's like the mutation primes them to really listen and react strongly when that interferon signal arrives. That enhanced sensitivity seems to be the engine driving this whole effect.
Okay, so we have these supercharged, hyper-responsive macrophages acting as a better APCs. What does that do functionally? What about the T cells they're supposed to be activating?
That's the functional payoff. They tested this directly. TET2 mutant macrophages were significantly better at priming naive CD8 plus 2 cells in lab experiments. Priming means essentially teaching the T cells what to recognize and attack. And the T cells that were primed by these mutant macrophages, they weren't just more numerous. A higher percentage of them became activated, showing markers like CD4414 plus $2. And crucially, these activated T cells produce significantly higher levels of granzyme B.
Granzyme B, isn't that one of the main weapons cytotoxic T cells use to actually kill target cells, like tumor cells? So the macrophages aren't just activating more T cells, they're activating T cells that are better armed, more lethal.
Exactly. More activated and better equipped. It's a double whammy. And they confirmed this back in the mice, too. T cells pulled out of the tumors from the TET2-mutant mice after ICT treatment showed a greater capacity to produce granzyme B. Interestingly, these T cells also showed significantly higher levels of PD-1 expression.
Hold on, PD-1, I thought high PD-1 on tumor T cells usually means they're exhausted. Isn't that what PD-1 blocking drugs, a major type of ICT, are designed to counteract? Why is high PD-1 a good sign here?
That's a really important point, and it can seem counterintuitive. In many chronic settings, like an established tumor, high PD-1 does correlate with exhaustion. But here, in the context of these highly activated T cells that are also pumping out lots of granzyme B, the high PD-1 is interpreted differently. It's seen as a marker of strong and recent T cell receptor, TCR activation. Basically, it means the T cells have been heavily stimulated by those super APCs. They're engaged, they're seeing the antigen, they're activated, not necessarily exhausted yet, they're seeing the signal strongly.
I see. So in this specific context, high PD-1 plus high granzyme B equals highly activated and engaged rather than worn out. That makes sense. Okay, that really seems to connect the dots. Mutant macrophages, hypersensitive to IFNγ, better antigen presentation, more activated, better armed T cells. Let's bring it full circle now. Did this detailed mechanism hold up when they looked back at the human tumor samples?
Yes, absolutely. They went back to the human NSCLC tumor data, specifically from patients with TET2-mutant CH, and looked at the gene expression patterns within the tumors using bulk RNA sequencing. The results mirrored the mouse findings remarkably well. These human tumors showed signs of increased infiltration by M1 macrophages, that's the pro-inflammatory fighting kind, and consistently more CD8 T cells present.
And the critical IFN pathway, was that signature there in the human tumors too?
Very clearly, the human tumors with TET2-mutant CH showed higher expression of key genes involved in the IFN response, like IFNγ itself, and chemokines like CXCL9 and CXCL10, which help attract T cells. They also looked at something called an IFN score, which is basically a combined measure based on a set of 18 genes known to reflect a T cell inflamed environment. And yes, that score was significantly higher in the TET2-mutant CH group. It confirms that these tumors have that hot, inflamed microenvironment that immunotherapy generally works best in.
So the human data really validates the whole chain of events seen in the mice. TET2 mutations in the blood lead to these supercharged macrophages in the tumor, making the tumor environment more responsive to IFNγ, leading to better T-cell priming and activity, and ultimately better survival outcomes with ICT.
That's the story the data tells. It really highlights how crucial the cellular context of these CH mutations is. TET2 primarily affects the myeloid lineage monocytes, macrophages. Because these cells are key players in so many solid tumors, this mechanism might well apply much more broadly than just lung and colorectal cancer. It really reframes TET2-CH. It's not just a sign of aging or general risk anymore. In the context of ICT, it's a specific, positive, predictive biomarker.
Which has potentially huge implications for treating patients, right? Instead of just noting CH is present, maybe we should be specifically testing for the TET2 mutation via a simple blood test. Could that help doctors predict who's most likely to respond well to ICT?
Potentially, yes. It could guide treatment decisions. Knowing a patient has TET2-mutant CH might increase confidence in using ICT, or perhaps influence the choice of specific ICT agents. It's also worth remembering, as you pointed out, that the mouse model success was particularly strong with anti-CTLA-4 therapy. CTLA-4 blockers are thought to work primarily by enhancing that initial T-cell priming step, which fits perfectly with the mechanism we've just discussed, the TET2-mutant macrophages acting as superior priming machines.
Okay, that brings us perfectly to our final thought for you, our listeners, to ponder after this deep dive. We've seen how TE2-mutant macrophages boost T-cell priming, which aligns well with how anti-CTLA-4 drugs work. But many patients receive anti-PD-1 therapy, which primarily targets already activated, potentially exhausted T-cells in the tumor. So the question is...
The question is, beyond just enhancing the initial priming, could these hyperreactive TET2-mutant macrophages be doing other things in the tumor microenvironment? For instance, could they somehow influence the T-cells later on, maybe making them more resistant to exhaustion? Or could they affect other immune cells? What else might be going on, influenced by this mutation, that could help maximize how well single-agent anti-PD-1 therapy works in these patients? Is the enhanced priming the whole story or just the beginning?
A lot more to explore there. It seems the connection between our aging blood system and our ability to fight cancer with new therapies is far more intricate and perhaps more hopeful than we realized. Thank you for joining us for this deep dive. We hope it sparks further curiosity about these fascinating links.